In today's video, I am going to take the 10th lesson which deals with cell cycle and cell division. In today's video, that is video number 1, I will be dealing with cell cycle exclusively. So, let us define a cell cycle. Cell cycle is a sequence of events during which the cell grows, duplicates its genome and eventually divides into two. So, once again, it is a sequence of events. So, cell cycle is a sequence of events during which the cell grows, its genome replicates or duplicates, genome means here is the DNA, duplicates or replicates, and finally the cell divides into two. This sequence, this entire sequence is known as cell cycle. It is broadly divided into two phases. The two phases are interphase and M phase. Duration of the cell cycle varies from organism to organism and from cell to cell. For example, in yeast, the complete cell cycle takes place just 90 minutes. Within 90 minutes, the entire cell cycle is completed. That is a case of a short cell cycle, but it may not be 90 minutes, it can extend to hours or days. Clear? Now, I am going to illustrate today a typical human cell which is representing a eukaryotic cell having a 24 hours cell cycle duration. So, let us take a 24 hours duration cycle. And when you take a 24 hours duration cell cycle, the longest phase of these two will be the interphase. Nearly 23 hours the cell will be in the interphase stage, and just 1 hour it will be in the M phase. So, that lengthy is the interphase. Nearly 95% of the cell cycle is the interphase stage and just 5% is the M phase. So, if you take a 24 hours cell cycle, 23 hours will be the interphase and just 1 hour will be the M phase. So, this is the cell cycle which is represented by a pie diagram. Now, let us start off with the interphase. The interphase has subphases. They are G1, G1 stands for gap 1, S stands for synthesis, and G2, gap 2. So, the three subphases coming under interphase are gap 1, synthesis, and gap 2 phase. Let us see what happens in interphase. Interphase, though it is called resting phase, the cell is not at rest, it is actually metabolically active, preparing for cell division. Look at G1 here. What is happening to the cell at G1? During this phase, suppose this is a cell, I am depicting the cell like this small circle. What happens in G1 is the cell is growing. So, it grew, became big. See the size? It has become big. So, in G1 phase, what happens is growth of the cell takes place. Now, it goes to S phase. S phase, what happens is growth stops and the DNA within the nucleus replicates or duplicates or doubles. So, the same size, I am not going to increase the size, same size in the S phase, but the difference is, or what is happening here is the doubling of the DNA. If I am representing DNA as this 2 here, so it is here 2, when it comes here it doubles, so I am giving the number 4. Or need not represent like that, I can represent in the form of chromosomes. Here there were 3, it will remain 3 here, but here when it reaches S phase, the size does not become big, the growth stops, and what happens to the number of the DNA? It doubles. So, there are 3 chromosomes, I'm going to make it 6, 1, 2, 3, 4, 5, 6, 6 chromosomes. Clear? So what happens in S phase is replication or doubling of the genome. Genome you can talk in context to DNA or chromosome. I've drawn here indicating the chromosomes. So we started with a cell with 3 chromosomes. In G1 it still remains as 3 because nothing is happening to the chromosome number, only the cell is becoming big. See the size of the cell, from this size it became so big. So growth is occurring in G1. Then here same sized cell, there's no further growth, but what happened? Doubling of the chromosome number. It was 3, I put 3 lines, it became 6 lines. Next, after S phase or synthesis phase, we are again moving to G2. So as I told you, what happens in G1, here also it's happening. Here again the cell grows bigger. See, the cell has become, so remember G actually indicates gap, but here you can remember what happens in G, growth. G for growth. Okay, the cell grows with and continues with the chromosome number. What was the chromosome? 1, 2, 3, 4, 5, 6. The chromosomes are 6. Clear? So once again, the phases or subphases of interphase, G1 or gap 1, synthesis or S phase, and gap 2 or G2 phase. What happens in G1 is the cell grows in size, whereas the chromosome number does not, gets altered. So we start off with this as the initial cell, a small size cell with 3 chromosomes, 3 lines indicating 3 chromosomes. What has happened during G1? It has just increased in size and the chromosome number still at 3, 1, 2, 3 lines. Then in S phase what happens? The size, again no further growth. No growth occurs in S phase of the cell. So the same size cell, see this is the size, the same size is here, no growth in the cell size. But what happened here? Synthesis of the chromosome number or doubling of the chromosome number. So I drew 3 lines, now it becomes double, 6 lines I drew, which is indicating the chromosomes. Then that is happening in synthesis. So DNA synthesis occurring. Then we are moving to G2. G2, as I told you, growth, further growth. So the cell grows further with the chromosome number. Which is the chromosome number? It continues with 6. Okay. Now it is ready for division. So this is how we say that interphase stage is actually a metabolically active stage of the cell where the cell is getting prepared for cell division. Now it is getting prepared to get divided. Okay, so we'll move to the M phase. So M phase is actually division phase. So as I indicated here, M phase, I am going to divide it because M phase first is followed by division of the nucleus. Division of the nucleus is called karyokinesis. So the nucleus first divides. Only after the nucleus divides, the cytoplasm divides. So it is followed by cytokinesis. Now karyokinesis involves 4 stages. They are prophase, metaphase, anaphase, and telophase. And after telophase, it moves to cytokinesis. So once again, now the cell is ready to get divided. It has become large with double the number of chromosomes. So what happens is first the nucleus within the cell divides. When the nucleus divides, that 6 chromosomes get divides, segregated to the 2 nuclei. So this can be represented, karyokinesis, if I represent it symbolically, see that large cell came, see in the initial size of the cell was like this way, then in G1 it became big, then what happens in S phase, it is still same size, but the chromosome number has increased and it has become bigger. So we started with 3 chromosomes. Okay, so 1, 2, 3 indicated by rods. Then 1, 2, 3 in G1. Okay, then what happens in S phase? The 3 becomes 6. 1, 2, 3, 4, 5, 6. Now what happens in G2? The cell grows in size with 6 chromosomes. 1, 2, 3, 4, 5, 6. Which means now the cell is ready to get divided. But here, how is the division? First, it is the division of the nucleus followed by the division of the cytoplasm. The 6 chromosomes are inside the nucleus, so the nucleus divides. And still the cytoplasm is not divided. Now what happens when the nucleus divides? The chromosomes get segregated, distributed to the 2 nuclei. So 3 will come here and 3 will come here. So this is representation of karyokinesis. See, karyokinesis is the division of the nucleus and distribution of the chromosomes. Here I started with 6 chromosomes, so the 6 chromosome gets segregated, 3 will go to one nucleus, 3 will go to the other nucleus, so they get equally segregated. Now what happens? The cytoplasm constricts and divides. Okay, so this is going to constrict and divide, and the cell is going to be divided into 2. So constriction took place. See, this constricted, and finally it separates. We get 2 daughter cells. 1, 2, and look at the chromosome number. Retains the initial chromosome number 1, 2, 3, 1, 2, 3. Ready? I hope this cell cycle, which is represented schematically, is also clear. So this is the initial state of the cell. Initial cell. Now it's entering interphase. When it enters interphase, the first subphase is G1, where the cell is growing. See, it has increased in size. See the growth. Then what happened? S phase, no growth, same size cell, but the chromosome number doubles. See, from 3 it became 6. Then next we are going to G2, again the cell further grows. Here I have indicated the nucleus, because very important to the next phase we are moving is M phase, which first is the karyokinesis, division of the nucleus. So therefore I do the nucleus. So the nucleus gets split into 2 nuclei and the chromosomes gets distributed. 3 goes to one nuclei and another 3 goes to the other nuclei. Meanwhile, the cytoplasm is also getting constricted. Now we see a completely deepened and constricted cytoplasm, but the 2 cells are attached and with the chromosomes separated in 2 different nuclei. And finally, cytokinesis. This is indicating cytokinesis. The constriction is further deepened and divide the cell into 2. And finally, what's the resultant? We get 2 daughter cells, and look at the chromosome number, becomes like the initial parent cell, which is, we started off with 3. And then if required, this can again enter into interphase if it's required to divide. Clear? So this is a brief outline of cell cycle. So look at the cell cycle. It has 2 phases, interphase and M phase. As I told you, interphase is longer than the M phase. It's very long. For a 24-hour cycle, it nearly lasts for 23 hours, and just 1 hour is the M phase. Now interphase has subphases G1, S and G2. What happens in G1? The cell grows. What happens in S? The chromosome or the DNA doubles, and G2 again the cells further grow. So now the cell is ready to get divided. It has become big to get divided. Now we are entering the M phase, the short phase. But M phase is further divided to karyokinesis and cytokinesis. Karyokinesis, first it is division of the nucleus and followed by the distribution of the doubled chromosome. They get equally distributed into the 2 nuclei, daughter nuclei, and then it is followed by cytokinesis. Now the process of karyokinesis is elaborate. It involves 4 stages which we will be learning in cell division. They are prophase, metaphase, anaphase and telophase. During these stages we are going to see how the chromosomes, how are they getting segregated and distributed to the 2 daughter nuclei. The way they are getting segregated is all going to be dealt in detail when in the next video, which deals with the events occurring during prophase, metaphase and anaphase, telophase. Clear? So I hope you have understood cell cycle. So I am repeating the definition to wind up. Cell cycle, now if you put the definition, it's applicable to the events that was explained. So cell cycle is a sequence of events during which the cell grows. See, the cell grows, replicates or duplicates its genome and eventually divides into 2. See, I've picked the points and given a tick against those spaces. So when you define cell cycle, so once again if I define, you can see all the events are covered in that definition. So it's a sequence of events during which the cell grows. So it involves G1 and G2 growth. Then the cell replicates or duplicates its genome number, which indicates S phase, and eventually, finally, they divide. And what's the end product of division? Eventually divide to form 2 daughter cells. So that is meaning the M phase. Clear? So once again, winding up once again with the definition of cell cycle. Cell cycle is a sequence of events during which the cell grows, duplicates its genome, and eventually divides into 2 daughter cells. And the 2 phases are interphase and M phase. Interphase is further divided into G1, S and G2, and M phase is divided into karyokinesis and cytokinesis, and karyokinesis is divided into 4 stages: prophase, metaphase, anaphase and telophase. And after that only they step into cytokinesis to form the 2 daughter cells, which is exactly similar to when we started the cell cycle. Clear? And always remember, cell cycle is not constant. It varies, the duration varies from organisms to organisms and from cell to cell. Thank you.